There is 4 components in fluid mosaic model.
Plasma membrane is a membrane that encloses a cell, separates the cell from its surroundings. Function of plasma membrane is separate the contents of cells from their external as a selective barrier that allows sufficient passage of oxygen, nutrients, and waste products, involved in signal transduction and involved in cell-cell recognition. Plasma membrane is fluid mosaic model. Fluid means that phospholipids and extrinsic proteins can move laterally along the membrane, while mosaic means that embedded proteins within the phospholipid bilayer creates mosaic appearance.
Phospholipid is an amphipathic molecule containing both hydrophobic and hydrophilic regions. Phospholipids composed of a polar head hydrophilic region and a nonpolar hydrocarbon tails hydrophobic region.
The fluidity of the plasma membrane depends entirely on its hydrocarbon tails. Unsaturated tails have kinks that prevent tight packing, which keeps the membrane fluid and moving. On the flip side, saturated tails pack tightly together, which increases viscosity and makes the membrane more rigid.
The plasma membrane relies on 2 main types of proteins to manage critical cellular tasks. Integral proteins are permanently embedded within the membrane, typically spanning the entire phospholipid bilayer. Because they cross this barrier, they feature hydrophobic internal regions that align with the lipid tails and hydrophilic ends that face the watery environments inside and outside the cell. Functionally, these proteins serve as transport channels or pumps to move molecules across the membrane, and they act as receptors that catch chemical signals from other cells. In contrast, peripheral proteins are only temporarily attached to the membrane surface. Instead of cutting through the lipid layer, they bind gently to the exposed heads of lipids or to the edges of integral proteins on either the internal or external side of the cell. These surface proteins provide crucial structural support by connecting to the cell's internal skeleton, and they coordinate fast chemical reactions by serving as enzymes or signaling assistants.
Carbohydrate chains only on the outer surface of the plasma membrane, usually branched oligosaccharides with less than 15 sugar units. Function in cell-cell recognition. Glycoprotein. Carbohydrate chain covalently bonded to protein. Glycolipid. Carbohydrate chain covalently bonded to phospholipid.
Next, let's look at the last component of the plasma membrane in animal cells: cholesterol. Cholesterol plays a vital role as a fluidity regulator. Its main job is to stabilize the membrane structure by restricting the excessive movement of the phospholipids, keeping the whole boundary stable yet flexible. At relatively warm temperatures, cholesterol will restrain the movement of phospholipids, making the membrane less fluid so it doesn't fall apart. At low temperatures, it does the exact opposite. Cholesterol blocks the phospholipids from packing too tightly together. This action will maintain membrane fluidity and stops the phospholipids from solidifying or freezing up,